Thank you for inviting me to share our study and our experience in African country. My topic is about when mission malaria met magnetic tropical diseases. My name is Tsai. I'm from the National Taiwan University, Department of Public Health, and also the Institute of Environmental and Occupational Health Sciences. Today, my work is about the vector-borne infectious diseases. So the outline will be the um, uh, vector-borne infectious diseases under the concept of the One Health. So in the beginning, we will talk about the One Health. And the second part, I will talk about the mission malaria, because we have mission by our government to collaborate with the African country to beat the malaria. So I will share with you some of our experience. During the malaria mission, we also found some fever of unknown origin cases. So we try to use the sample there and collaborate with local researchers. We try to find some magnetic tropical diseases in Sao Tome and Principe. I will talk about the different vectors, including the fleas, mosquitoes, and ticks, and also the mites. Most of the diseases was the first documented in this country. And finally, I will have some summary. Okay, so under the One Health concept, you will know that it's integrative thinking, integrative health risk management perspective, which acknowledge the systematic interconnection of humans, animals, and also the environmental health. So it's a big concept, and we not only focus on human beings, but also the environment, the plants, and also the animals. So by the One Health initiative, you can find that more than about 70% of vector-borne infectious diseases or zoonosis are caused by the vectors or the vertebrate animals. From the figure, you can see, for example, the avian flus, tick bite fever, or the West Nile, chikungunya, Ebola, Lassa in African countries. So a lot of the new emerging and the re-emerging infections appeared. So when we talk about the vector-borne infectious diseases, you should know that vector is important. Humans and also the natural host, pathogens. The most important is the environment. We should know the environment, then we can do some intervention to change, to stop from the disease infection. Usually when we talk about the infectious disease, the vectors around us, we will ignore their existence. For example, we will suffer from the biting from the mosquitoes, from the ticks, flea, and louse, and also the mites. But if they can infect people, then human will get diseases. Then we need to treat. Then we will know what kind of medicine can be used. About the tick-borne diseases, just an example, you should know that most of the pathogen can transmit it to next generation by trans-ovary transmission. It means that the ticks will transmit the rickettsia or the pathogens to the next generation. So based on this concept, we will not very hard to control them if we do not use the chemical pesticide. So it should be considered about the environment, how to control to management the environment. Okay, so it's a One Health initiative. When we control the environment, we should consider about all the livings in this area. And combined with the goal from the United Nations, they said that sustainable development goals. In this figure, you might understand that the three about the health, most of the work is forward the goal to the health, human health. According to the 3.3, the world says that by 2030, we want to end the epidemics of the AIDS tuberculosis, malaria, and also the neglected tropical diseases. So usually I combine words. You can see the first is the ATM, the three top infectious diseases, AIDS, tuberculosis, and the malaria. The ATM is a money catcher, right? So if we suffer from the severe diseases in this country, we must pay a lot of money to face on these diseases. And also the magnetic tropical disease is the NTD. In Taiwan, it's a new Taiwan dollar, also mentioned about the money. So if we can control and if we can improve the infection in a country, then we will save a lot of lives and improve the economic development, and then we can earn a lot of money to improve our daily life. So it is important when we do the disease control or the vector control, they will fulfill with the goal of the Sustainable Development Goals. So in our previous experience, we have a malaria mission in this country, Sao Tome and Principe, in the Gulf of Guinea. Here, combined with two islands, Sao Tome and the Principe Island. So our mission, Taiwan is here. Usually we will take the flight from Taiwan to Thailand, to Germany, and then to the Portugal, then to Sao Tome and Principe. Usually we will spend 3 days, including the travel times. Okay, so it's the place we want to visit. In this area, we want to control the malaria because the infection rate, the incidence rate is really high, almost about 45%. So in this country, we try to focus on the human part and also the vector part. In the human part, we have a headquarters and have a laboratory. We try to train people to do the proactive surveillance. We provide the rapid test. We provide the treatment, the drug, and the medicine. And then we also set up clinic especially for the malaria infection cases. So after the work in this country, we try to figure out the potential infection people in the village and also in the kindergarten and all the village. So the risk population include the children under 5 years old and also the pregnant women in the village. So after 10 years, we have a lot of steps to cover the disease. About the vector parts, in this country, we want to know what's the major vectors responsible for the malaria transmission. So we set up many light traps to collect the mosquitoes. So we also train the local people, local researchers to participate in this work. So we collaborate together and we try to figure out what's the source. We found a lot of mosquitoes, anopheles mosquitoes in the source. And we also, during the epidemics, we try to train the local people to use the chemical pesticides, alpha-cypermethrin. The method is the indoor residual spraying. And we also try to record all the mosquito species responsible for the disease transmission, including the chikungunya and the Zika and also the yellow fever or the dengue transmission. So that's the mosquito part we done in this country. To our surprise, we used to say that the malaria vectors in this country is the Anopheles gambiae. But by the molecular analysis, we found that the sequence is very special and very unique. So by the construct of the genes of the CO1 genes, we found their phylogenetic tree, the morphology is very different. You see that the Anopheles is a new mosquito. We named the Anopheles cluzii. So these mosquitoes is the major populations in this country. Used to, we said that it's a mosquito, Anopheles gambiae M form mosquitoes, but now the name has been changed. not only the mosquito name changed, but also we know their behavior is really specific and very different because the mosquitoes never rest inside of the house. After they take the blood, then they will fly out of the house. So we try to fulfill with the behavior of the mosquitoes and try to set up new control strategy. So from the photo, you can see that we collect the mosquitoes and to evaluate the efficiency of the indoor residual spraying efficiency. So one people is staying inside of the house and one is outside of the house. We found that a lot of mosquitoes appeared in outside of the house. So it's the first data. And we also try to analyze the haplotype of the CO1 sequence. So it's a very beautiful photo. You can see that in Sao Tome Island is the gray color accumulate together. Then is the Principe. Then is the mainland African country. It include the Mali and also the Cameroon and also the Kenya. So it's just like a population isolation and have a speciation of the mosquitoes in this country. So from now on, we name the Anopheles mosquitoes in this country is the Anopheles clusia. So by the time goes by, we found that the incidence rate decreased. You can see that the first step, we used the indoor residual spraying to control the mosquito density by using the chemical named alpha cypermethrin. And after we found the mosquito is very different, so we tried to shift to modify the control strategy. We use the larvicide. The name is the BTI. BTI have two different formation, the BTI 200 and the BTI 3000. We try to apply them and to evaluate the efficiency after we use. We found that the BTI 200 is still very good for local people to spray. So you can see the incidence rate correlate to the mosquito density. The case decreased from the 44% to the 2016 years. It's about nearly to the 1%. So it's a very good data for us because a lot of people were saved. So it's a control strategy and efficiency in this country. However, we found that some very special situation, because you can find this figure. Most of the people, they can free of the infection of the malaria, but we found that the case of the fever is still high. So it means that something we don't know still ongoing, still happened in this country. Okay, so while incidence of malaria has decreased, the number of the fulfilled illness remains. So we try to consider about the environment. What happens in the environment? Is the fever caused by the vector-borne infectious disease or the foodborne infectious diseases or some kind of that? So we try to figure out the questions and try to provide some intervention methods. The first is that we found local people. Most of people, they lack of the shoes and they barefoot to walk to the school. And we found that some of the people was infested by the flea. Also, me and our Professor Lian and also our co-workers there also infected by this fleas. We call the tangiasis. The tangiasis local is it's a very special tick. They live in your soft tissues on the foot. So it's a tangiasis. So we try to figure out this kind of situation and alert the local public health agency, and to let the local people knows we should have shoes to protect our foot. And also they need to clean their environment to kill the flea larvae in the environment. With the time goes by, we found that the malaria has been decreased. The tangiasis, the cases decreased. However, we found that still a lot of vectors very active in our environment. For example, the ticks and other fleas from the cats. And also we found the rodents with some kind of mites. So we try to think about whether the rickettsiosis appeared in this country, whether the dengue fever or the chikungunya severe still appear in this country. So under the project of the malaria control, we try to use the same materials to analyze the sero preference. So we use the serological study and also the molecular study to prove whether they exist in this country. Okay. So the first part is about the mosquito part. To our surprise, because we living in Sao Tome and Principe for 6 years, and we found that not only the Anopheles mosquitoes, but also the Aedes mosquitoes. So it's very reasonable to correlate with the Anopheles mosquito and the Aedes albopictus to the disease transmission. Based on the reference, we found that near the country, they have an outbreak of dengue. But in this country, the relative reference is very limited. So I asked my students collaborate with the local researcher. We try to analyze the antibodies against the dengue infection. You can see that the prevalence of the antibody positive rate is very high, about 35% in local. So it shows that not only to control the number of mosquitoes, but also the Aedes mosquito is also important because all the residents, all the people living there, they will have the water container to save the water. You can also find some Aedes mosquitoes living in the containers. So it's a source. We should to do some intervention to let the local people knows the risk. So that's the first part about the mosquito diseases. And later on, we also found that some vet medical doctor, they told us that they was bitten by the ticks and get a fever. So we correlate to the tick-borne diseases in this country. We try to analyze whether the spotted fever group rickettsiosis appeared in this country and also the Q fever caused by the infection of Coxiella burnetii in Sao Tome and Principe. So here shows the country further. The map is the Sao Tome Island. It's the Agua Grande. It's the metropolitan city in local. And here is the Principe. Based on the concept of the structure of the malaria control, we have a lot of human samples from here. So we have some improvement by the local headquarters, by the Department of Health, and they agree that we can do this kind of study. You can see that in local, a lot of goats. That's a very important situation in local. And we collaborate with the local vet medical doctor. They've tried to help us to figure out what kind of ticks hover on the goats. So as you can see that a lot of embryoma ticks, including the variegated. If in Europe or in other African countries, the embryoma variegated responsible for the tick-borne diseases, including the African tick-bite fever. So it's a good point. Maybe we can do the analysis whether they carrying this kind of pathogens in this country. So it's the first sample we collect in this country. The second is that we collect another ticks named the Embryoma astrium. It's a very popular, and according to the reference, this kind of species only appeared in this country. So maybe it's a very special species in this country. We collect all the ticks from the cattle and also the dog. Okay, so it's the vector specimen. So after we bring this specimen and back to our laboratory, we try to extract the DNA and also do the PCR analysis. So we found that The first is the variegated, about the 81% of the tick samples with the Rickettsia antigens. And about the astrium, about the 22, about the 52% with the Rickettsia species DNA. But none of them with the Coxella burnetii. However, we found a very special DNA similar to the Coxella burnetii. We call the Coxella-like endosymbiont. It's a kind of Coxella burnetii, but whether they are pathogenic still unknown. Okay, so that's the first data we know. Okay, in this country, have the vectors, have the pathogens. Whether the people was infected and caused the fever, still unknown. So we analyzed the DNA and also the sequencing all the PCR products. We found that there's a novel strain of Rickettsia africa in this country. And we also found the sequence of the Coxella burnetii, the like endosymbiont in the ticks. In order to know whether the local people was infected, so we do the serological analysis in this country. So you can see we use the antigen of Rickettsia africa and also the Coxella burnetii to test. Finally, we found that the seropositive rate, the criteria is about 1.64. So we found the positive rate is about 8% against to the spotty fever, and about 6.7% against to the Q fever antigens. So that's the first data to show that the spotty fever rickettsiosis may be happen in this country, and also the Coxella burnetii infections. Okay, so that it's the tick-borne diseases we first identified in this country. So the photo shows the immunofluorescence assay. We call the IFA to detect the antibodies against the rickettsia. And we also use the Western blotting. We named a soft Western blotting to differentiate the specific antigen, including the Rickettsia connori and also the Rickettsia africa. So I think the data is very solid to prove. So later on, we are waiting if any people who get a fever and search for the diagnosis that we can confirm the case from the real peoples. When we're living in the South Tome Principe, we usually will see the eagles flying in the sky, and you will see that a lot of rodents very active in our environment. By this kind of image, if you live in Taiwan, you will suffer from the infection of the scab typhus. It's named the mite-borne scab typhus. So we have some idea, maybe we can find very similar to the Orangia pathogens appeared in this country. So we try to use the serological analysis to explore whether the people was exposed to the Orangia tsutsukamamushi or similar pathogens. So you can see the table. We use the ELISA, we use the immunofluorescence assay, IFA, and also the Western blot. We found, yes, they did. We did detect the antibodies against to the outer membrane protein. Okay, so this is the first data proof that the antibodies in African country. The positive rate is about 5.8%. But it is very important because you know that the scab typhus only appeared in Asia area. We call it a triangle area from the Japan and also the Korea and to the North Australia and to the Arabia area. So it's a triangle. So after search further reference, we found that some occasional case was reported in African country. So it's the alerts that maybe some rodent traveled with some traffic and bring the ectoparasites, the mites, and transmit the pathogen around the world. So it's a important finding for a lot of us about the vector infectious diseases. Okay, so summarize my talk today. In Sao Tome Principe, the main work is to do the malaria control. Based on this concept, we also analyzed the mosquitoes, including the Aedes mosquitoes. We found that the dengue maybe happen in this country, and also we found also the fleas, Tonga penetrans, the fleas, and also the Cytinocephalides felis. These two species of fleas in this country may cause the two diseases. The first is tongiasis. The second is the flea-borne rickettsiosis. We name TRG, transitional group rickettsia group. So this is also recorded in this country. And about the chicks-borne, the spotty fever groups, the several evidence had been set up. And the scrub typhus also been recorded. So based on this finding, we collaborate with local people. We try to help to document all the possible vector-borne infectious diseases in local. So in the future, if they want to set up a national or country control strategy, they should consider all the vectors, not only focus on Anopheles mosquitoes. They may focus on mosquito part, the flea part, the ticks, and also the mites. So that's all the work we done in Sao Tome and Principe. And I know that some similar diseases also prevalence in different countries, in African countries. So if in the future, we can collaborate and we can try to help to do some vector control to help local people to improve their daily life and daily qualities. So it's my brief introduction about our work today. So thank you for your attention and welcome to visit Taiwan to see all the beautiful sites in Taipei. Thank you. Thank you very much.